I'm a CCMU scholar, grad scholar, ATRGV, and so I'm looking at specifically here focusing on stomach contents with red drum. Okay. So just to give like a little background on importance of looking at food webs and stuff like that. So food webs do give us an understanding of the interactions between different trophic levels, the transfer of energy between these levels and the organisms, as well as, exchange of matter. And it also gives us an idea of the role that a certain species will play within the ecological community. And for this, we're going to be looking at the top of the food web. So this understanding leads to better conservation and restoration efforts and increased jobs and economic value to local fisheries. Specifically here, we're talking about South Texas and red drum, which is a big recreational fish down there. Okay, so juvenile red drum, like I mentioned, is the target species for this project. So they are seen as a predator fish within the estuary. So they do use the estuary as their nursery system. So juveniles will stay there and live and feed until they hit maturity around their 3rd or 4th year, and then they'll eventually go out into the Gulf to live in adulthood. When it comes to their diet, they're usually seen as opportunistic feeders, but seasonality, size, and location can all probably have an effect on their diet. So that's kind of what we're looking at here. First, just a quick brief of the objectives. So first is to compare the trophic web between two different sites, which we categorize, one as a lagoon system and one as a bay system, among 4 seasons. Or we broke it into 4 quarters throughout a full year. And secondly, to reconstruct the trophic web using stable isotopes, so carbon-13 and nitrogen-15, and again, both these sites. So just to give a background of where we're looking at, so Texas, because it's huge. We are all the way at the bottom there where the arrow is pointing down in the lower Laguna Madre. So if we zoom in, we're going to find where the 2 sites are. First, it's called Holly Beach, so you can kind of see the orange, it's a little bright in here, color. So it's more of a sandier bottom. This is the one that we categorize as a lagoon system. It's a lot more open to the lower Laguna Madre. Whereas our second site down below is called South Bay. That's our bay system we have. It's kind of repetitive, but so that's more closed off to Laguna Madre, much more protected. Has more of a muddier bottom, and there's a higher presence of seagrasses, which you can even kind of tell, maybe with the lights off a little bit better, but the bottom one, South Bay, is a lot darker colored than the top one, so you can kind of tell those differences already. Okay, so getting into the methodology for all of this, as for field collection, we just followed all of Texas Parks and Wildlife protocols, so just fish just like they did. So on the left-hand side, we have our gill net set up, and then on the right-hand side, we have us pulling bag stains for both the prey items and trying to get the smaller juvenile sizes. And like I mentioned, it's a full year of data and broke it up into 4 quarters. So we did start going out in October of 2022, and that first quarter went through December. The second quarter started in January of 2021, went through March. Third, starting in April, went through June, and then the 4th quarter started in July, went through September, so we got that full year in. So more on the field collection. This is more for the stable isotopes just due to cost, but there were 10 fish per site per quarter and 2 non-overlapping size classes. For the gut content, we had a lot more samples because that doesn't cost us money. So there are 5 fish per size class. So we had a small size and a medium size, the small being 20 to 40 centimeters in total length, and the medium size being 50 to centimeters in total length. So talking about what we did for our content analysis. So all of the red drum that were either that small or medium sized were opened up, and then the gut contents were taken out, and any of the prey items that we find in there were retrieved and identified, and then they were tallied and categorized, and to use that data to calculate the percent index of relative importance or percent IRI via 3 different metrics. So I have a number percentage, weight percentage, and frequency of occurrence percentage. And to calculate the IRI, because I don't have it on here, just for reference, it's the number times the weight divided by the frequency of occurrence. Just a brief overview of the process for the stable isotopes. It was just to extract the white muscle tissue, mostly dorsal fin if we're talking about fish, for both red drum and the prey items. And then all those tissue samples were dried, ground up, and then sent out to University of Arizona to get those signatures. Okay, so now looking a bit into the results more specifically. So juveniles inhabiting both locations and in both sites. Oh, I just saw that. Both size classes, sorry, do have similar diets, generally just being a bunch of crabs and shrimp, which you can see on the top part. But there were some slight differences in prey types, which thinking is potentially just related to prey availability, given what is available in the bay during a certain season. So looking at the sites, so this is going to be the number percentage. So it's basically just like abundance of what's found within the stomach contents themselves. So I have South Bay and Holly Beach comparing. So this is going to be all 4 quarters together. So you're going to see Panopidae, which is mud crabs, followed by Pinedae or shrimp, in South Bay being the top 2 most abundant. And then once you start getting into the smaller ones is where you start seeing some differences. So here we have snails coming in 3rd, whereas in Holly Beach, again, we're seeing that same trend with the mud crabs followed by the shrimp. But then it changes to kind of like the Cephalus or blue crab as that 3rd most abundant prey item. And then, like I said, I used it all to calculate IRI metrics as well. So we have that for South Bay and Holly Beach, which it's important to look at all of them because depending on a certain fish, some of the like abundances can be skewed and show that like, oh, we only eat a certain thing, but it could have been like one fish having like a feast on one type of prey. So just one thing to point out here is vegetation. So in South Bay compared to Holly Beach, you are seeing a higher frequency of occurrence and number percentage than Holly Beach, which is kind of going along with what I pointed out earlier, where we're seeing a lot more vegetation just present in there. Vegetation covers just some seagrasses, so like Halibut, Tilapia, and just some algae. So now looking at it by sizes, with all 4 quarters, again, we're seeing those same general trends. The colors are all going to be the same, so you don't get lost. So we have mud crabs, shrimp, but then we have the stargazer fish. So this is one of the things that I was talking about. So there was like a small fish, 2 like small fish in quarter 4 that just had like 50 of these tiny fish in their stomachs, which is pretty wild. So that's where we're seeing that difference. Then we have mud crabs and shrimp again for the medium size, and then followed by blue crab again. So looking at the IRI metrics for both sizes, again, you're kind of seeing the same general trends. I think to point out again is vegetation. You're now seeing it occur a lot more in the medium size than you are in the small size. And then fishes, again, that's changing slightly with more of that frequency of occurrence in the medium size than the small size. So if we're going to break it down based on the quarters, we're going to see, again, the same general trends with shrimp and mud crabs. But actually, so for quarter 1, 3, and 4, shrimps were the most abundant, followed by mud crabs. So that's actually a little bit of a flip from the other patterns we were seeing. Whereas quarter 2 is, again, you see that way larger portion with mud crabs followed then by shrimp. For quarter 1, we actually have Lagonorrhoboides, or pinfish, being the 3rd most abundant. Quarter 3, we're seeing snails again. Quarter 4, we're seeing a larger portion of blue crabs. Oh, sorry, quarter 3. Quarter 4 then is that fish that I was talking about. So we're seeing that difference. And it's a lot of, that's on one page, but, and so you can kind of see all of them together. So we have our IRI metrics for all 4. And so the crabs are kind of the biggest difference we have here. With Q2, it's really high on all, oh, it's come up on the bottom. Well, okay, that's fair. It's a lot higher number percentage, weight percentage, frequency of purge, and IRI. Whereas you're going to see that difference between the different quarters there. So taking the quarters into consideration, Redding and Anderson. So it was significant, but as you can see with the R value, it's 0.323 if you can't see it. It's pretty low. So it's just showing the percent similarities based on the IRI by quarter. So you're kind of seeing that they're all pretty similar. You're not having that high similarity, with some variability in that Q4 portion, but also just no significant differences in the groupings, and that was from with the SIPR. And then just very quickly, for the stable isotopes portion, this is mostly focused on the gut content. This is just preliminary results, so it's just Q1. So that means the 10 fish per site, 5 per size class. But you're just kind of seeing similar trophic webs on both size classes and both sites. So it's pretty interesting to see. The blue up there is going to be that red drum. And it's then followed by the respective prey items. So you have the mud crab, which I showed as important, followed by shrimp, and then 2 different fish species. So just overall to conclude, finding that both these sites do have similar diets, and we're seeing that again with the size classes as well, with some small variation between size classes and quarters, but again, they tend to be eating the same thing. And oh yes, thank you, NOAA, for this, and everyone else in the lab.
We got time for several questions.
Okay, I think so. Go ahead.
The fish in the Q4 that you're referring to, I think about greed of man, but are there any behavioral studies that, or you know of any behavioral studies that have been done on that fish that may explain greed when it comes to consuming? You mentioned the 50 fish.
Yeah, that I'm not too sure. I haven't really looked at behavioral studies. Everything that I've looked at when it comes to diet, it's just kind of they eat whatever is there. So it might have been that they just came upon, because they were all really tiny too. So it might have just been like a school of fish that they just came by and swooped in and got it. But yeah, it was
Kind of like the
weird.
whale goes through.
Yeah, exactly. Yeah. Go ahead.
So regarding vegetation showing up in red drum's diet, do you think that's a result of them directly preying on the seagrass, or is it more so when they're like trying to get to the crab and the shrimp hidden in the grass, some of that vegetation ends up in their stomach?
Yeah, that's a good question. So yeah, I think it's just like bycatch, because they are like pretty much eating like all of the benthic species, I think like shrimp, crabs, and stuff like that, and even like the fish that they're eating kind of stay below. It's also like I didn't mention, but these sites are really shallow. So like you go in there and like it hits like maybe your knee or something, and I'm not that tall, so it's probably even shallower for everyone else. So yeah, so I think it's just bycatch because they're going down in there.
Thank you.
Yeah.
Just a quick question. Did you guys do isotope signature for the detritus?
No, but we will be looking into that sooner. But yeah, because this was just like the Q1 very quickly, but yes.
Great, thanks.
Yeah.
Do you know another isotope question? Do you
Yeah.
know the lag time for stable isotopes in white muscle? And then how do you plan on matching that up to the gut content analysis?
Yeah. So like, that. Oh, okay. Sorry. Okay, there you go. I got it. Yeah, so that's like with this, it's kind of interesting where we're, some people pointed out, because they're very close to their own prey items that I have pointed out here, especially like the 2 fish that are here. So that the signature should be showing up once we get the next quarter in.
Okay.
So yeah, I think.
So it'll be like a quarterly lag.
Yeah,
time,
so
you
it should come in after. Oh, this is the mic, sorry.
All right, one last question, or if not, I'd like to give a round of applause to all the